This podcast was produced by OAR FM Dunedin with support from New Zealand On Air.
I'm usually in the studio. Myth-busting and de-extinction. What does it mean to you? Welcome to the OI podcast, the Otago Institute podcast, conversations with experts from the worlds of arts, sciences and Mataranga Māori. Brought to you by the Otago Institute, the Otago branch of the Royal Society for Arts and Sciences. Kia ora, nau mai, haere mai. For those of you new to the OI podcast, welcome. I'm Dr. Barb, a quantitative ecologist with a background in modelling, climate change, biodiversity, conservation prioritisation and SciComm, and a PhD in botany from Otago Whakaihu Waka, the University of Otago. Today's show, episode 135. I can't believe we've got to 135 already. Unbelievable. We're talking about myth-busting, de-extinction and ancient environments and ecosystems with Associate Professor Nic Rawlence, the winner of the 2025 Prime Minister's Science Communication Award. Congratulations, Nic.
Thank you. Thanks for having me on.
Yeah, no, it's fantastic and great to have someone from our very own University of Otago here, right here in Dunedin, as the winner of one of these amazing prizes.
No, it's lovely to be here. Still very amazing and humbling award. Still very, very much sinking in and very glad that I no longer have to tell little white lies since I've known about this since mid-March.
It's a funny little thing while they get all their ducks in a row and decide when is the opportune moment to release the information.
Yeah, very much so. I was just thinking of a few weeks ago, I was in a airport shuttle with Veronica Meduna, who I work with on The Conversation, who was asking me, did I know anything about it? And I just had to swear, black and blue, no, I know nothing. Nothing to see here.
But they did have some big ceremony in Wellington, didn't they?
Yeah, no, they did. So they flew my wife and I up to Wellington, and I got to bring some guests along. So my brother and his wife and some supporters and collaborators from Te Papa and Victoria University and teachers I work with from the biology educators of Aotearoa, New Zealand. And we had a ceremony at Parliament where we were presented with the trophy, which is deceptively heavy. It's like trying to pick up a meteorite, and a big glass frame certificate, and gave an acceptance speech.
Fabulous. And did you actually get it presented to you by the Prime Minister?
Yes, I did. And
I guess you don't get to choose your Prime Minister, but still.
No. Got presented by Christopher Luxon, and we had the Science Minister Penny Simmons there as well presenting.
Of course. And today we're going to talk about some of the things that, well, I guess most of the things that you got that award for, because it's not a standard kind of research award, right? It is the communication prize, Science Communication Prize. Yeah. But I will just go through, because obviously you are a scientist by trade. So you have an undergraduate degree and a Master of Science from the Victoria University of Wellington in New Zealand, but a PhD from the University of Adelaide in Australia. What on earth possessed you to go to Australia for your PhD?
I was looking around for PhDs and adventures, and at that stage, Alan Cooper, who was a New Zealander doing ancient DNA, had been coming through New Zealand doing a speaking tour. I was looking for PhD students, and I really got hooked, and I went and did a PhD over there and worked with Trevor Worthy, who's a famous New Zealand palaeontologist who was doing his PhD there at the same time. And it was very much the questions I wanted to look at, the species I wanted to work on, like working on moa and how did they adapt to climate change and humans and
climate change there we're talking about ancient climate change, right?
Very
so
much.
Like ice ages.
Yeah, think of the Ice Age movies. So this is like climate change stretching back.
Wow, I mean there's a little bit to be.
Yeah,
I wouldn't take the Ice Age mode just while we're on myth busting. Yeah. Let's not propagate the myth that the Ice Age movie is verbatim.
But yeah, we're looking at like climate change going back 10, 20, 30, 40,000 years. And at that stage, there just wasn't the scientists in New Zealand doing that. There wasn't the funding to do that.
Sort of research, I guess. There's little pockets of it because it's quite expensive doing ancient DNA.
It is.
Compared to, well, I guess it's got cheaper over time everywhere. Yeah. DNA stuff's all got cheaper because everybody's got, I don't know, new ways of doing it that are faster and you can do things in bulk and whatever. Yeah. But apart from the whole cheap, like everything in the DNA sphere getting kind of cheaper, ancient DNA is a
very
whole level
expensive.
of more expensive on that, and more difficult as well, I guess, because you're using stuff that's scarcer to start with,
so
because the material kind of degrades over time, I'm imagining.
Yeah, no, it very much does. So the really good analogy is like if we think of modern DNA is like the Auckland Yellow Pages, if that anyone remembers that. Really,
gosh, that's a really long time ago, isn't it?
really, really thick 2-volume. If you want to go and work on the engineering page, you can find that page, you can photocopy it, you can transcribe it. It's really, really easy. But if you take that Yellow Pages and you put it in a wood-fired pizza oven at 500 degrees overnight, it comes out as crumbs, fragments, shards. And that's what we're dealing with ancient DNA, really small fragments, easily contaminated with modern DNA, like the DNA from us or the curator eating a chicken sandwich and then touching moa bones, for example. And so...
And I mean, even people finding it in the first place.
Yeah.
And I guess also there's like, if it's just, I mean, a lot of moa bones, for instance, they're in caves, right? So they're kind of not buried deep. They're, or, people find them because they've kind of been pushed out of the ground, whether it's by a flood or some erosion event or just someone exploring in a cave. And all of those, I guess, instances are possibilities for it to get contaminated.
Yeah, pretty much. And bones are very much like a giant yellow kitchen sponge. They're really good at soaking up all the DNA from the surrounding environment. So as the DNA of the moa degrades over time, it's soaking up all the environmental DNA, all the bacteria, the plant DNA, the DNA coming through the water that's flowing through. And so the DNA you're after is teeny tiny minuscule amounts. And we had to work in these very specialist labs.
How do you get rid of the modern DNA?
You can't necessarily get rid of it. We're to use systems that preferentially targeted the endogenous DNA, so the DNA that belonged to the bone we were interested in.
How does it know what bits belong to the bone?
So,
I mean, the bone itself doesn't have DNA in it, does it? It's just minerals. Or am I wrong?
So there's bone cells called osteocytes that have got the DNA, and they're trapped within this calcium matrix that's your bone. And so what we want to do is decalcify that bone. So we chop out a bone sample, we grind it all up. We put it in EDTA, which you also find in shampoo. It strips out calcium from the bone. So
if you've got like hard water that's got like lime.
Pretty much. Yep.
Just basically if you're from Oamaru and you've got limestone water.
Yeah.
It's all that white stuff that collects around the kettle.
Yeah, so EDTA will strip out that. It'll decalcify your bone, and then you end up with something that looks like a snow globe.
Oh, nice.
That you can shake up. It's decalcified bone, and then you digest all of that. You break apart the cell walls. You extract all the DNA. And then there's this technique invented by a guy called Curry Mullis, who got the Nobel Prize and retired and promptly went surfing, called polymerase chain reaction. And so simply it's this technique that allows you to photocopy these tiny small bits of DNA.
That's what we know as PCR, isn't it?
PCR, yeah.
Yeah, right.
Photocopy these tiny small bits of DNA to workable concentrations. Now, to photocopy that, we can design these really specific little tags that bind to your DNA or your engineering page, and only that.
Yeah.
And that'll allow you to photocopy all of that DNA up and not all of your contaminating DNA.
So, I just... How do you know that that's the stuff that you want? Like, how do you know what to put the tag on?
Usually there's been a lot of, in the case of moa, there's been a lot of research that's been done in the background, like characterising, say, the mitochondrial genome. So this is the genome that's passed down through mother to daughter, or the nuclear genome. So we have a very good idea of what the picture on the puzzle box is. So we can go in and design very specific tags just to get these bits based on the DNA sequence.
But then you have to know the thing before you find the thing.
For moa, we knew what we were looking for because of all the work done beforehand. If we are looking at something brand new, so a good analogy is macrocania, which is this giant extinct camel tapir-looking thing from South America. So it's about the size of a camel with this kind of like pseudo-trunk. No one really knows what it's actually related to, and you haven't got that nice grounding to start with. So you actually have to go in with these universal tags, which will bind to all DNA of vertebrates, for example, and then hope that when you do all the lab work and the sequencing, you can then out the other end go, right, let's throw out all the human contamination, let's throw out all of the environmental contamination, and we've got something that's actually non-human mammal that we think is our species of interest. It's a lot harder if you don't know the evolutionary history or background of the animal you're looking for.
Right, so you're sort of like building a lot on what other people have already done. And
Very much standing on the shoulders of giants.
And I guess that's why it kind of, like, you hear about the same key species again and again and again. Like, you hear a lot about mammoths.
Yeah. And
I guess with mammoths, they've got... And the dire wolf thing, just going back to just picking out a few there. So you've got the moa.
Yeah.
Moa really recently extinct. So you've got a lot of bones. You've got bones.
Well preserved.
Well preserved, reasonably recent ones. So they're not like ancient. They're, hundreds of years rather than tens of thousands or hundreds and thousands.
And we at least know we at least know there are birds. So if we start getting bird DNA and it's not coming up as the common contaminants like chicken.
Right.
And it's... When you compare the DNA...
How closely related is a moa to a chicken?
Oh, you're looking at tens of millions of years of divergence.
Yeah. Because they're like tropical bird...
Yep.
Chickens, rather. Yep. Are like tropical birds.
So you're looking at least 60, 70 million or more years of divergence between a moa and a chicken.
That was slightly off out of left field, as they say. Yeah, just kind of to get an idea of where we're looking, because I mean that's
Yeah.
Whereas something like, what's the nearest relation to a moa that's still alive?
A tinamou. So it's a small chicken-sized flying bird from South America.
Right.
And you've got to go back 60 million years to get from a tinamou to the common ancestor of tinamou and moa.
Oh, wow. And so that's still quite a long time, isn't it?
And then you've got to go back 65 million years to get to the common ancestor of a moa and an emu.
Right.
And then you're going further to get back to the common ancestor of a moa and a chicken. So back into deep time before the extinction of the dinosaurs.
Yeah, before the extinction of dinosaurs. Wow, that is quite amazing. Yeah. So I guess it's sort of like the more recent stuff is Gondwana land, isn't it? Is that what we call Gondwana land?
Yeah, so we're looking, Gondwana started breaking up hundreds of millions of years ago through to the last bits that separated were like South America from Antarctica and then Australia from Antarctica and around about the 30 to 40 million year range. But it started breaking up over that time period. Like Zealandia, New Zealand, started separating from Eastern Gondwana in this very zipper-like fashion down in the south. Started about 83 million years ago and then didn't actually finish until about 55, 60 million years ago, where the last land connection between New Caledonia, which was part of Zealandia, and say modern-day Queensland finally severed.
Finally separated out. Wow. Yeah, I mean, all of these things. It's quite strange, really, because it's like big numbers,
huge numbers,
and it's like it's quite hard, I think, for I mean, even, we're sort of working in science, but it's still quite hard to think of those big numbers, like 65 million. It just sort of rolls off your tongue. But like if you write it out, you've got 65 and then 6 zeros. That's a lot of time. And when you compare that to, the whole of the amount of time that humans have been, like the human species, as a, what we would recognise as someone of the same species as us, what's that?
250,000 years for anatomically modern humans,
Yeah, and that's, compared 250,000, so that's a quarter of a million compared to 65 million. They're very different.
It's huge numbers and mind-boggling to get your head around. There's a great science communicator friend of mine, Professor Flint from Adelaide, who's...
Do they work on stone?
His name's Michael Mills, and his alter ego is Professor Flint, who's a paleontologist.
Right. Okay.
And he had this for Paleontology Week at the South Australian Museum when we lived in Adelaide. So it's this 10 days just dedicated to science communication and paleontology. He had the toilet paper of time, and you get this roll of toilet paper and just roll it out and go, that is deep time. And then anatomically modern humans is this tiny little shred of the bit of toilet paper he's holding, and all of evolutionary history is just spooling down the floor.
Yeah, and I think it sort of gives you pause to think of your the kind of the small place of humans in the world. It kind of gives you a recalibration of the importance of humanity and, the amount of time that we think things are. I mean, people will say, I've heard, you hear ridiculous things like, that has to be a road. It's always been a road forever.
Yeah.
And you think, yeah, but actually we've only had cars for like 100 years in New Zealand. It hasn't always been a road. It's like very recently been a road. And before that it was a bush, and, it's had a long time before it was a road. And there's that kind of framing things back into our life cycle, which is actually even our life cycle. We lived quite a long time in terms of, most animals and insects. Do you know how long the average moa lived?
It took, for some species of moa, it took about 9 years to reach adult maturity. So there are these things called lines of arrested growth or lags in moa bones, very much like tree rings in trees. And
you jumped ahead of me because the next question was obviously going to be, how do you know that? So,
And so we think it takes, for some species, about 9 years to reach adult maturity and then another couple of years to reach sexual maturity. And they're very slow breeding, so they're probably living for quite a long
It's a quite
time.
long-lived animal. I mean, in terms of birds, that's quite a long-lived bird. It's not those cockatoos and parrots. Some of the parrots are amazing how long they live, eh? But, still quite long-lived and then quite slow
breeding.
Because if you're that big, you have really big eggs. You don't have a whole lot of eggs.
So you're probably laying one at most 2 or 3 eggs, depending on the species and how valid analogies or comparisons are to other ratites like emus and cassowaries and ostrich, but you've got a very long-lived, slow-breeding bird. And there's actually been some ecological modelling done that there's no way you could sustainably harvest moa. It's just one of these species that whenever humans turned up in New Zealand, whether it was East Polynesian arrivals or Europeans, any level of harvesting would have just been too much for the population or the species to handle, and they would have gone extinct.
Crazy. And have we found like nests of moa nests with eggs in them, or just moa nests? Because, presumably they're ground nesting, right? Because they're flightless birds.
Yeah, no, Jamie Wood, so a good friend of mine who did his PhD in the geology department with Daphne Lee. I think he finished about 2008 now at the University of Adelaide. His PhD was reconstructing what central Otago was like prior to human arrival, and in some of the rock shelters where he was finding coprolites, so desiccated moa poo, he was actually finding moa nests where you've got your feathers, you've got eggshell, you've got your coprolites, you've got all your twigs. They've been clipped at the angles that match moa beaks and everything.
I mean, that's quite cool, isn't it?
It is very, very cool.
Do you know how big they were, these moa nests?
I'm
guessing quite big, right? But then again, there was all sorts of different sizes of moa. Because we talk about moa like it's one species, but actually, what is the generally accepted 9 species?
9 species, and you got everything from the size of a goose or a turkey up the very top of Northland, which would look quite cute on your back lawn, right through to the South Island giant moa that could reach up to 3 metres height when it was browsing or going for
and
food.
That's similar to the one there's the model of it in Tahura at the front there with the little egg that's got the money donation thing.
Yeah,
so
and then if you go up to the top floor of Tahura, there's the old school reconstructions where you've got the giant moa all feathered out, and then you've got this really squat looking one that is called the heavy-footed moa that Beverly McCulloch, a famous New Zealand archaeologist, described as a 40-gallon drum walking in a little kid's gumboots, or as I tell my students, Cartman out of South Park.
Oh, yeah. But presumably didn't swear as much.
No, and probably didn't eat cheesy poops either.
So the big one... Was that not in the North Island?
No, it was. There were two recently diverged species. There was a North Island giant moa and a South Island giant moa, and it was the females that were the biggest. And so that display in a Tiger Museum where you've got the male and female skeleton is kind of looking at the extremes of let's take the smallest male and let's take the biggest female in the collection and line them up.
Right.
But the differences within population, so within Otago or North Country, wouldn't have been as extreme.
Still, but walking around, like, what did you say? It was 3 metres tall.
3 metres tall when it would have had its neck
When it's had
right
its
up.
neck high up. And I guess they walked around sometimes with their necks high up. But yeah, like a 1 metre 60 kind of person. That's twice a person's height.
Yeah.
It would have been quite phenomenal to come across that in the bush. Because were they bush, the big ones, were they bush or were they sort of inland plains, that kind of
the giant
grassland?
moa were very cosmopolitan. They were across all habitats, from sea level to alpine, from open to dense forest.
Right. And I guess they didn't have that many predators, probably the big Haast eagle.
No, they would have had Haast eagle for a lot of the really big ones. And then for some of the smaller moa, there was also Isles harrier, which was the largest harrier in the world.
Yeah. But not like the little kestrel that we have now, the little New Zealand falcon.
No, that wouldn't have taken.
That's too.
Maybe would have taken out some of the small chicks of some of the smaller
babies,
species. But not the adults.
And kahu, the Australasian hawk, that's not just recently introduced, is it? That was always a bit cosmopolitan between here and there.
It was always a bit cosmopolitan, but the kahu
expanded its range a lot lately.
Expanded its range into New Zealand only within about the past 400 to 500 years.
All right, so it didn't overlap with the moa.
After the extinction of
Haast
eagle and Isles harrier.
Have we got really good dates for when the last of the moa went extinct, or when we think they went extinct?
We've got some good dates. It's very hard to try and work out when the final moa passed off its mortal coil.
Yeah. Because there might have sort of just been little pockets that dribbled and drabbled on.
We've got some radiocarbonation I did of some crested moa remains from up Mount Owen. So this is up in northwest South Island. It's, if you've watched the Lord of the Rings movie, it's where they come out of the mines of Moria into that limestone moonscape is Mount Owen. And we're looking at dates and that'll be one of the last natural moa of the late 1400s.
Right.
And so you're probably looking at extinction of moa by about 1500. But there would have been.
So we're sort of thinking Tudor England.
Yeah. So you would have had functional extinction, which probably occurred quite quickly within about 100 to 150 years, and seems to have happened across the country at the same time, regardless of what species you are, upland, lowland, population density, and then you would have
big species, big moa, little moa,
little moa, and your last natural individuals would have been up in the alpine areas, so it would have been like the likes of the giant moa, upland moa, which is your like mountain goat of the moa world, and crested moa. And you would have just had these little
When you say
pockets.
Crested moa, what kind of crest did it have? Was it like really special?
We're not sure. If you look at the top of the skull, there's actually a whole lot of pits for feather insertion.
So it could have been like really, really like
we've never
mohawk
found
type.
Possibly like a little mohawk. We've never found those feathers, but enough that it probably did have some sort of crest.
Crest, okay. Just want to imagine that there's this. I mean, what kind of plumage? Like they're always depicted as being sort of browny coloured, sort of kind of kiwi coloured, kiwi feather coloured. Were they brown? How do we know that?
We've got.
Sorry, I've turned this all into moa.
Oh no,
my
it's all
God.
good.
Sorry,
we've got really nicely in central Otago, because it's really quite dry, as we get a whole lot of feathers actually preserved, and there seems to be actually two sorts of feathers, and so we've got the drab brown ones that give you kind of like the mottled kiwi plumage, but then we've actually got ones that are the bottom 2/3 are actually quite dark, and the top is white, so it actually gives you a really speckled plumage, more like a chicken. And one of the I did as part of my PhD is we were developing this technique to see if we could actually get DNA out of feathers. So like if you're doing a modern DNA project, you'll pluck a feather and then there'll be a little skin tag that comes with it. And that's pretty much what most people got DNA out of, and they never tried to get DNA out of the rest of the feather. So we developed this technique that could dissolve all of that feather, extract all the DNA, and then you could go back to the fossil record and work out what these feathers belong to. And so we will find actually both these different plumage types, you are actually getting some species, and again it was based on a small sample size of what museums would let us sample their feathers and chop bits off. But you could get this quite mottled Kiwi-type plumage and restricted to some species, but you could actually get other species that have this very speckled plumage, and then some species that actually have both. So what we're not sure of was that...
You don't know if it was males, one colour, and females.
Male, female, juvenile, adult, or different plume, the mottled brown, camouflage, or the speckled one on the same individual, but there was definitely some quite cool plumage variation going on in moa. It was still very camouflage because...
Right.
Animals or birds that arrive on islands tend to become more drab compared to their, say, mainland relatives.
Oh, really?
So if you think of a takahe is, even though we call it a purple chonky rainbow chicken, it's more drab compared to a pukeko that's just arrived here from Australia in the past 400 years.
Yeah.
But also moa had to be camouflage. The top predator, Haast eagle, was a bird that hunted by sight.
Right.
It's why Kiwi camouflage.
That was so big, though. I mean, really, how did the Haast eagle mainly live off moa? Was that like
moa and a lot of medium-sized birds? So not only moa, but it would have gone after kakapo, takahe, your adsbill, which was about a metre high, what we call killer tank of a rail, the same family as takahe.
I was just thinking that
goose.
And the extinct goose, yeah. I was just thinking though that like moa, like the big moa, did their size not give them some protection? I mean, like
it did give them some protection. Like
I imagine they could give quite a whacking kick.
They could give a kick. I've always wondered with koiwi, ancestral human remains that are discovered in New Zealand.
Yeah.
Or that are unearthed, is why there aren't more with blunt force trauma from being kicked by a moa.
Maybe the moa just didn't realise that people were dangerous.
Quite possibly. They were quite ecologically naive as well.
Yeah, because that's a thing with New Zealand birds in general, like recent nowadays birds. Like they've been here with people for quite a long time now.
Yeah. So there would have been some protection given their size, but some of the smaller moa would have been targeted, including the chicks. But what we do find with moa is the ones that really get stuck in swamps.
Oh, yeah.
So they would have got stuck in swamps.
And that must give you quite good, like, bones and DNA and stuff.
Quite good bones, quite DNA, especially if those swamps have got basement limestone rock underneath and they're buffered by limestone runoff.
It's all in the chemicals of that kind of environment that they're in, isn't it?
Yeah.
Stop the decomposition.
But those moa, their pelvies, or their pelvis.
What's a pelvis?
Pelvis, just
like
plural
pelvis.
for is. And I've
it's a good Scrabble word, isn't it?
No, it is. It is a good one. I've excavated 10 to 14,000 year old moa swamps in North Canterbury that you pull up a moa pelvis and flick it over, and you get this big ridge coming up the pelvis. And on either side of that ridge, if you punch through the bone, is the kidneys. And so if you think of that big giant ridge in the pelvis, and I'm for the listeners, as I'm miming a moa pelvis and then Haast eagle talons, as the Haast eagle would have come down to this moa, stuck in the swamp, and then it just come in with its talons and gone like that. Into the back of the pelvis and then just gouged out. And so you just these massive puncture marks and then gouge marks where they've gone after the moa that are stuck.
Right. So it's quite interesting though, as you can see sort of who's killing who.
Yeah, no, very much so.
Yeah.
You can see these behavioural interactions that actually really preserve in the fossil record.
Yeah, no, it's interesting. And I guess you're saying that they're quite cosmopolitan in the type of environment that they were in, the big ones.
Yeah.
And I guess some of the smaller ones were more constrained to forest areas?
Yeah, so Little Bush moa was strictly very much a forest species. You've got eastern moa, which is one of the most common moa in the South Island, was very much restricted to that lowland swamp forest. Upland moa, the mountain goat moa, was in very rock areas, so all down the Southern Alps and bits of Central Otago, the Catlins, this more mountainous area.
Yeah. Hey, I'm going to move on because we are halfway through the interview now, so I thought I'd move on to this whole de-extinction and myth busting because, of course, that's a big part of why you got this award. And I thought I'd just pose a few questions and just like
fire away.
I mean, people talk about the dire wolf and the mammoth, but I thought just because we've been talking about the moa, one of the big things that people throw at the de-extinction, I mean, I don't know. Part of me thinks that it would be really cool to see a moa, like a real live moa.
It would be cool.
Or like a whole herd or flock. I
Yep.
don't know if they're a herd or a flock, actually. There must be a flock. Birds are flocks, aren't they? Even if they're walking.
Stampede of moa.
Stampede of moa. Yeah, honestly, I have to think that out. There's a dictionary somewhere of the
The names for all
the
the
names
groups of
for all the
birds.
groups
Yeah.
for all the birds. Anyway, we'll just go with flock. If there was, I don't know, I still think it's a herd of
Herd,
moa.
yeah.
It would be cool. Do we know if they lived in big groups or if they were more individuals? Because I mean, that's a kind of social behaviour of birds, isn't it? Some of them are in big flocks and some of them are quite individual and come together in pairs. Or
I think they were more the individual or small family groups.
And what makes you think that?
Comparing to other ratite species where you're looking at emu and ostrich, where it tends to be small groups as opposed to really large herds. But we know in moa that it was the males that actually incubated the eggs. So
How do we know that?
So again, with ancient DNA, as it was work done by David Lambert, who's at Griffith University in Brisbane, where if you extract the ancient DNA from the outside of moa egg, you only get male, which is the incubator.
Right, okay.
You don't get female. And if you do it from the inside of the egg, you get male or female, which is the chick. So it's the male that's actually doing the level of parental
So it's just
care.
just sitting there, it's just kind of its DNA is rubbing off on the egg.
Yep, and just absorbing into the egg.
Quite crazy, isn't it? It just makes me feel like I'm just sitting here on this chair. My DNA is rubbing off on the chair while I'm
Pretty
sitting
much.
here. It's crazy, isn't it? It's very Gattaca. Why are all these people in Australia looking at our native birds? Why don't they look at their native birds? I mean, there's like a lot of birds have gone extinct in Australia as well. Not even just birds, but marsupials.
Part of it is a lot of these people started their careers in New Zealand.
Right.
So Alan Cooper and Dave Lambert were working in New Zealand, and when people were overseas, they keep up the same research interest. And until very recently, Australia's been almost too hard to do ancient DNA. It's
Why?
just you've got to go back further in time to get to, say, the big charismatic megafauna. It's a lot hotter, it's a lot dry, not that conducive to ancient DNA preservation.
But you were just saying that Central Otago was great for conservation because it's dry.
It is for bone.
Oh, right.
And feathers and eggshell, but if you try to get, say, DNA out of the coprolites,
The poos. Fossilised poos.
the success rate is going down the gurgler. And we think that's actually because you've got that, they're a lot more porous than bone, and you've got that wild swings from baking hot summers to permafrost winters that just screws with the DNA. So
right.
as technology has increased, or ancient DNA technology has increased, we're now beginning to be able to push the envelope back a lot further in time and have a better understanding of what conditions preserve DNA in Australia quite well. So it's like Kangaroo Island in South Australia, or southwestern WA, or parts of Tasmania, really being able to target these sites. So ancient DNA in Australia is beginning to... You can do a lot in the Holocene, so the past 10,000 years or the past 5,000 years, but pushing that further back in time is really dependent on the sites and the techniques that you're doing.
So it's more that a lot of our species went extinct
very
more recently.
Yeah.
Right, so that's like a big bonus in terms of looking at extinct things because they're less degraded because it's...
Less degraded, we're
'cause
more
there's less
temperate, we're not as hot and barren as Australia.
Right. And I guess also the tropics where it's like really wet and hot and humid, that must degrade things really fast.
Really rapidly. And you're really looking at the past 1,000 or 2,000 years, and you're really pushing the proverbial uphill to try and get anything out of it. You've really got to target.
And I guess that kind of... does that kind of distort what people are working on to working on bigger things? Just because they're bigger, they're more likely to have more DNA in you basically if you're bigger because you're bigger.
They're bigger, more solid, or not necessarily more DNA, but you've got.
I mean,
more,
you do though because you've got DNA in every cell, and if you've got more, if you're bigger, you've got more cells.
You've got more bone to play with. It's more solid, more likely to preserve DNA. But also the big things are the sexy things that get funding.
Right.
And what we're finding with some of the stuff we do is actually the really small, like little obligate. Small birds and lizards and geckos that are telling all the quite amazing stories as well. So the Lilliputian biodiversity.
When you say obligate, what are you meaning there?
Species that are really tied to their environment. So if you think like a bush wren or a Duversel's gecko or a forest skink, of like these birds, these animals are really tied to the environment. So they're really good proxies for what the environment is doing through time. Have just as amazing stories to tell.
And you think that they're really tied to the... I mean, what gives you the information that they are really tied to that specific environment? Because is it the environment or is it some specific mutualism in that environment or is it just that you've only found it from a particular place because it's quite small and it's hard to find?
If we take the bush wren, for example, it's where all the specimens were found in the 1800s and early 1900s till they went extinct. They were only forest species. They've got very limited flight capacity. So they're a good proxy for kind of like what the forest was doing. So if the forest...
I'm just wondering if we're just making a just so story about the bush wren because they went extinct quite recently and maybe pre-human they were actually really widespread because the environment changed so much over
They
that
were
time.
quite widespread pre-human. Like if you look at the Holocene, they're spread around the South Island, but when you actually reconstruct using, say, fossil pollen or fossil plant macrofossils, so bits of leaves, twigs, cuticles, seeds, if you reconstruct those environments around where those bones were found, they're all forested. So if we try to work out what the environment was like, we try to look at say like a time slice where you're not getting major environmental change. So where were they in that time slice, and then does that pattern hold true if we say go to 20,000 years ago, so the height of the Ice Age, and where we're finding bush wren bones then are a lot of them around Honeycomb Hill in the West Coast, where it was this nice forest refugia.
Right.
So some species are very much tied to their environment, and some species don't care what environment they're in. They're very, like the giant moa, very cosmopolitan. They can move.
Right. They're all over the place. Yeah, it's interesting though, isn't it? It all goes back to the plants.
Pretty much.
Yeah. So do you feel quite vulnerable there, being so dependent on all the plant people?
We need the plants to reconstruct what the environments are doing. We've got really good plant records going back, like pollen records and plant macrofossil records going back through time with all the, say, the amazing work, for example, like Marcus van der Goes from GNS is doing.
I was thinking that's why places like Folden Mar are so important.
Yeah, definitely.
Because they've got that really thin layers where you can tell that time change and really great preservation of various macrofossils, and then
we need that because that's like kind of like direct evidence of what the environment was like, as opposed to say isotopes, which is a bit more derived or indirect evidence.
Let's not get sidetracked into isotopes today's shows. But I was thinking back on the myth busting. So I guess there's a lot of talk about de-extinction because there's a lot of people who, I mean, it's a funny term really, de-extinction, because like extinction is a thing that's gone.
Yep,
it's
gone
extinct.
forever.
It's gone forever. But it's interesting because I just grew up in New Zealand, obviously, same as you and most of the listeners probably. And extinction is, like, we talk about extinction here because New Zealand has one of the highest rates of extinction in the world. And, you know, we're not looking like we're slowing down at the moment. No. And then being in conservation, ecology and stuff, we talk about extinction, so it's very common. And then when I was in the UK first doing postdocs, like as you were doing in Australia, and people were talking about butterflies going extinct, but they had this thing called locally extinct. And I thought, that's this very strange idea. And what they meant by locally extinct was something like a blue butterfly that would go extinct locally in the UK. So it was going extinct in a kind of human geographical political area, but of course it's not extinct. It's still, you know, like
still there.
It's still all over Europe. Like there'd be things that were like, you know, basically the UK was the very edge of their range,
Yeah.
and they were, basic Europe, right across Asia. Europe was their natural range, and they were very much still present.
Yeah.
And they were going locally extinct. And so there was this kind of, and I was doing mapping and stuff, and it was all kind of theoretical and where things were and how they were changing and how things had changed with current observed climate change, which is really interesting because people were sort of like, oh, is climate change working? And I was like, yeah, well, because at the time, and this was like, I don't know, 15 years ago now, we were modelling recent observed climate change and observed species distribution changes that were modelled by that climate change and then predicting into like the current
future,
now.
yeah.
Well, no, just predicting into the current. Like, so we had atlases from the 30s and 60s, and then the climate change, like the recent climate change between then and the 80s, and then using that to make the model and then predict it into the current and then predict like the actual now, which was, 2004, 2006, that sort of time period. So it was like we were modelling now.
Yeah.
We were modelling into the future as well, but we could build the models on the past models, test it on current models, and then predict it into the future. And it's amazing how people are still like, oh, is it a thing? And I'm just like, no, of course it's a thing. Like, it's there, it's a thing. Like, just accept that it's a thing and then move on from there. But anyway, I digress. I was talking about local extinction, and it made me really aware that in New Zealand we have quite a firm idea in our heads of what extinction actually means.
Very much.
Compared to other places where they kind of have this kind of wishy-washy idea that it could come back, and because if something's locally extinct, you can,
reintroduce it.
You can reintroduce it because you know what its environment still is, because it's still still somewhere. It still has that environment. You can reconstruct the environment. Whatever you did to muck up the environment, you can just kind of wheel that back somehow, and you can do that in various amounts. And I wonder if that's played into this idea that de-extinction is possible.
I suspect it has, and it's. I think it's also if we're thinking of colossal biosciences, which is trying to do this de-extinction, I think it's
so
also.
This is like a big company from overseas.
Yeah, there's $10 billion valued biotech company in Dallas that wants to bring back creatures from the dead. I think it's also them playing into that to also sell what they're trying to do to...
so they're basically trying to make a Jurassic Park.
They're trying to make a Jurassic Park.
And I wonder how much the movies of Jurassic Park, how much that plays into the idea that all this is possible.
I think they're very, very much leaning into it. And they're going... You can't do true de-extinction. So
it's interesting though, isn't it? Because there's a lot of people that probably wouldn't care so much because it's so exciting. I mean, like in the movies, Jurassic Park, they do depict lots of people. Like it's like a zoo. It's like a theatre park. It's like there's roller coasters and whatever. And I mean, I don't know if that's what Colossal is actually presenting. What is their idea? Are they planning on having a zoo, or are they planning on releasing these things into the wild?
Their argument is they're doing this for conservation, to release these animals back into the wild to re-establish lost ecological connections. And so they're one of their arguments for the creating a genetically engineered, hairy, cold-tolerant Asian elephant and calling it a mammoth is they could release this back into the wild and it would stomp all over Siberia and turn it back into the productive mammoth steppe and sequester carbon and combat climate change.
Right.
But you would need thousands and thousands and thousands of mammoths to actually pull that off.
So I guess there's two things, isn't there? There's whether it's really de-extinction, whether they can do it, and then the kind of the proposed reasons that they say that they're doing it for.
Yes.
And if we take those separately and go, okay, can they really do it? So you're saying no.
No. Short answer is no. So
what are they? So what would they have? They would have something that is kind of looks like a mammoth. It might function like a mammoth.
We don't know. So if you were going to truly de-extinct something, you need to make a carbon copy.
Right.
Effectively, you need to do classical cloning, like what occurred with Dolly the sheep, the first cloned mammal that got cloned, I think, 30 years ago yesterday.
Yeah, it's a long time, eh?
Long time ago. And so the way cloning works is you need to take the genetic blueprint of an individual.
Yep.
and you, from a donor cell from a sheep, you then take another cell from a sheep, you remove its genetic blueprint, so it's just an empty sac, and then you put that genetic blueprint and the empty cell together, you give them a zap, they fuse like sperm and egg, they create an embryo, you put it in a surrogate, and after hundreds and hundreds ago, you get Dolly the sheep.
Right. So basically, you need the DNA.
Very long pieces of well-preserved DNA.
So you need lots of really good DNA, and then you just kind of put it in the cell, like as if it was there.
Yep.
But then the key part is that you then have to have something to incubate that.
You need a surrogate.
And if you're doing a sheep are still here, so you can just pick a sheep.
Yep.
And use that as a surrogate. And we kind of like from IVF stuff and human stuff, we can do that really quite.
Yeah, and it still takes hundreds to go. So there's only been one instance of true de-extinction, which was a subspecies of Pyrenean ibex, which had died out in the wild. Scientists had cryogenically frozen some tissue, and they did this cloning, and the animal lived for a few minutes before it died of multiple organ failure. But you need long strings of very well-preserved DNA, like hundreds of thousands, millions of A's, G's, C's and T's letters of genetic code long. You're not going to get that with ancient DNA. It's that Auckland yellow pages in the wood-fired pizza oven.
Right, so that's why the mowers really, and the thylacine as well.
Thylacine and the direwolf and everything. So what Colossal are doing is strictly genetic engineering. And so they're sequencing the genome of these extinct species. But
taking really recently extinct species. So no one's talking about Jurassic Park here.
No, it's very recently, like tens of thousands of years. Your DNA is still fragmented. So you can reconstruct the genetic code.
Right.
You can't reconstruct those long strings of well-preserved DNA.
Right.
So they'll take that genetic code, they'll compare it to a close relative. So if we take the grey, the scientists sequence the direwolf genome, they'll compare that to the grey wolf, the closest living relative, and they go, where in all of the 2.5 billion individual letters of code long is there a difference between a grey wolf and a direwolf?
Right.
Now, the grey wolf and direwolf are 99.9% identical, but that's still over 1 million individual letters of code difference.
That are different, right.
And they found the supposed differences, and they did 20 edits to 14 genes using a technology called CRISPR-Cas9, which is, think of it as a very precise pair of scissors that you can chop out a chunk out of the grey wolf genome and then insert a piece of synthetic DNA matching the direwolf genome into the grey wolf.
So what you're saying is they've taken a very small proportion of genes that they know are different out of a massive number that are different, because there's that 99.5 and that's like thousands of genes, but they've taken a very few of those that they think are the ones that are controlling something, or just that they know are there.
The ones they think are the important ones. So changes of big effect, ignoring all the ones that are changes of small effect that may be quite additive.
Right, okay.
And then so they've got that genetically engineered grey wolf genome.
Right.
And in this case, they then put it in one of these empty cells, gave it the zap, put it in domestic dog surrogates, and they got these 3 direwolf pups out the other end. And so you have something that in Colossal's view, it looks like a duck, quacks like a duck, talks like a duck, therefore it is a duck, and it will miraculously fill the job vacancy in the ecosystem and everything will be hunky-dory.
I guess, though, you could argue that they've started somewhere. So, like, at some point, because, I mean, genetic stuff historically moves really fast, right? So, okay, they've only got a small proportion of those. At some point, they could get all of them. You've got to start somewhere, so start with a few that you know are big changes.
For Mora, you're looking at conservatively 10 million individual differences. But even if you create, say, a poor facsimile of a moa, like a genetically modified emu, you've got something that looked like a moa, but we have no idea whether it actually functions like a moa. If we think of Colossal's chosen the giant moa because it's big, it's sexy, Peter Jackson wants a giant moa. But the giant moa is actually one of the most complicated ones to bring back, because some of the ancient DNA evidence we've been reconstructing diet, is we know there's sexual dimorphism in moa.
Right.
the females are bigger than the males, but we're actually seeing evidence that you're getting ecological niche partitioning. They're occupying different parts of the ecosystem between males and females and juveniles and subadults and teenagers and adults.
So there's two parts there, really, isn't there? There's like, can you make this thing?
Yeah.
So let's just
a mower.
Let's just imagine that they are at some point in the future able to identify all the different base pairs that have everything that's changed or mutated over that big change between when a mower and the nearest relative that they're using to match it up with, which is 60 million years.
Yeah.
So at some point they've got such amazing genetic stuff, and stuff in genetics is a mystery to most of the population, and it goes very fast. So they can, at some point, just identify all the things that have changed, all the things that have mutated, and they can actually reconstruct the entire genome, DNA, of, say, a mower, just for argument's sake. Then the next thing is getting something that can incubate it, because there's a whole lot of hormone things that happen.
Yeah, so with the mower, it's really difficult. So we don't have nice surrogates like direwolf can be used. A domestic dog as a surrogate.
Right, so you have to have something that makes the egg, right?
We have the whole chicken and egg, and this is where it starts getting very sci-fi, sci-techy, which brings in a whole lot of conversation about communities, including Mana Whenau's comfortableness with genetic engineering. We need to have these discussions. Is you need to pull this off, you have to, one, create transgenic chickens, and two, you've got to create an artificial egg. So what Colossal needs to do is they take their genetically modified emu genome, or arguments say their reconstructed mower genome. They then need to put it in what we call primordial germ cells. Now, that's a fancy term for the precursors of sperm and egg.
Right.
They will then take these primordial germ cells and put them into a developing chicken embryo that has been mutated to not produce its own sperm and egg.
Right. And
then that chicken will grow up and you'll get a whole flock of chickens that will look like a chicken. If you take a tissue sample from them, it will be genetically a chicken, but they will produce sperm and egg containing mower, in quotes, DNA.
Right.
That you would let chickens do what chickens do and mate and lay eggs, and then you would crack that egg open and then pour that yolk and that embryo into this artificial egg construct,
right?
To then grow up all of these.
Because the egg's much bigger of a mower than a chicken. So how do you get the yolk big enough?
So that's one of the challenges. So
I'm sure that
a giant mower egg is actually 80 times the size of a chicken egg. So even if you created a flock of genetically modified emus that produce the sperm and egg of a mower, you've still got an 8 times difference. And so a yolk is a single cell. It's actually the biggest cell in the world. And so not only do you have to delicately pour this yolk and this embryo and the egg white into this artificial egg, is you've got to add more egg white, but you actually have to inject more yolk into the yolk cell without it exploding.
Yeah.
And
I see. It starts to, the more you think about the actual detail of it, the more it sounds a little bit more complicated.
Because, and this is what Colossal doesn't sell. They sell, this is all possible.
Yeah. And I can sort of see that, because in the movies, everything is possible.
Yeah.
I can sort of see that. Yeah. And I guess the final hurdle is you get one and then you have the behaviour, what is learnt from the adults, which don't exist. So that's
and who's going to teach it to do all the mower things? And then also, is it going to function like a mower? One of the really good examples I use is there's some fungi in New Zealand forests. It's called ectomycorrhizal fungi. It's truffle-like fungi. So you go tramping through the beech forests, you get those purple, reds, blue fungi.
Yeah.
And they've got a symbiotic relationship with the plants that allows those forests to spread across the environment through time. Now, we know moa ate those fungi and spread them, but is that because they were a giant herbivorous bird that an emu could fill, or is that because they had their own unique microflora? Intestinal microflora.
Yeah,
so if you bring back a genetically modified emu, is it going to be able to perform the function of a mower?
Yeah, what you're saying is you need all the other things as well. You need the ticks and the mites and the
you need the entire ecosystem.
Bacteria of the mower.
So it comes back to Jurassic Park, the very first Jurassic Park movie, where that Triceratops got sick. You can't bring back the animals unless you bring back all the ecosystem, and then you shouldn't be surprised the animal gets sick because it's eating something that was never around when it was alive.
Yeah, no, it's quite tricky, isn't it? Especially with things that, eat plants.
Yes.
I think for some reason I think that like animals that eat animals,
A lot simpler.
It's simpler.
But even with the direwolves, there's Colossal has actually admitted that none of the hunting behaviour is innate. They kept saying, you don't need parents, it's all innate, they'll know how to be direwolves. And they're actually having to bring in a grey wolf matriarch from an outside pack to actually teach them how to hunt and be direwolves.
Oh, right.
So
interesting. Honestly, I do feel that the direwolves is a direct response to Game of Thrones.
Yep, and they could not make the genetic change in the grey wolf that would give them the pale coat colour like in direwolf, similar to a dingo or a coyote, because, excuse the pun, it was dire consequences. They made 3 other changes to other genes that gave them pale coat colour, but it was the public perception of a direwolf, which is out of Game of Thrones, which isn't surprising when I say that George R.R. Martin, who wrote the books, is the cultural advisor for Colossal.
Yeah. Can see that. We're almost out of time now, Nick. I'm sorry. I should have shifted on to the myth busting sooner. I guess. Yeah, we have to just leave all the rest of the questions to another time.
No, more than happy to come back.
So, hey, sorry about that, folks. We'll get Nick back on at some point. What we might do is ask for questions from the audience. Oh my gosh, that would be amazing, wouldn't it? We've got questions.
More than happy to answer those.
Hey, you've been listening to the OI podcast with Dr. Barb in conversation with Associate Professor Nic Rawlence. If you've particularly enjoyed this episode, delve back into our back catalogue and enjoy episode 129, Community Archaeology with Zach McIvor. Episode 106, Indigenising Archaeology with Rachel Wesley. And June 2024, Where Does the New Zealand Bird Fauna Come From with Michael Knapp, a colleague from the Zoology Department there. I don't even think I mentioned that you were from the Zoology Department. That was in the next part of the biography that I never got to. Sorry, Nick. And of course, we have had Nick on before, back in July 2024, Ancient DNA and an Extinct Duck, where we were talking about that Merganser. I have to say that right. Hey, thank you for tuning in to the OI podcast. Please do like, subscribe, follow, do all the things, and pass our name on to everyone, because word of mouth is our best. You've been listening to the OI podcast, the shout out for the arts, sciences and Mataranga Māori.
You can support this podcast by becoming a member of the OI. Check out the Otago Institute for Arts and Sciences on Facebook or our website.
If you'd like to listen again to this episode or others from the series, look for the OI podcast on OAR FM's website, oar.org.nz.
Brought to you by the Otago Institute for Arts and Sciences, the Otago branch of the Royal Society of New Zealand.
This podcast was produced by OAR FM Dunedin with support from New Zealand On the Air.